right.
All right, guys. So we are back for next lecture. So now we have with us Dr. Elvira Maria Saraiva. So let me quick introduce her for you. So she's currently a full professor at Universidade Federal do Rio de Janeiro. She's a biologist like me, and she did her master and PhD studies at the University of Federal University of Rio de Janeiro as well. Then she did a postdoc at the NIH under supervision of Dr. David Sachs, and she's a very renowned scientist with many awards and more than 100 papers already published. But more than that, you could also taste a little bit of Elvira yesterday, of how kind and nice person she is, which is much more important than any other title. So Elvira, thank you very much for being here with us, and the stage is yours. Thanks.
Thank you very much for the invitation. It's a pleasure to participate in this course. So I'm very happy to talk about this nice way of dying, that is the NETosis, that you can see in this first slide, that you are seeing a huge net, like a fish net, trapping parasites. I don't know if you see my arrow. It's okay?
Yes.
Okay. So you see one parasite here, another here, and here you see a flagellum of a third one. So, oops. It's not going. Halfway. So I'm going to talk about the 7 points. So beginning with the discovery and the inducers of this kind of death. It was first described in neutrophils, that is this cell here that you can see in the blood, stained with GIMSA. So you see the granules in the cytoplasm of the cells and the nuclei that is polymorphic. And neutrophils was a cell that was not much studied because, well, we knew that they are very active phagocytes, and they granulate, so they have many different mediators in their granules, and this can be released to the extracellular medium. And so people are saying, well, okay, this is it. It's the only thing that the neutrophils can do, and they do very well. But then in 2004, the group of Arturo Ziklinski, that is this person here, described this new mechanism. Actually, the mechanism was already new for different researchers, but all the time they are thinking that was an artifact. And what he discovered is that upon some stimuli that we are going to see later, neutrophils can release to the extracellular medium its chromatin that you can see here stained by DNA and stones, and also with different proteins from the granules and from the cytoplasm, and here is represented by the elastase. And more than that, what is very interesting is because this structure can trap in this work different bacteria that you see here, the nets holding and trapping the parasite, and also it has some microbicidal activity. So if they incubate the neutrophils treated with cytokalasin D to inhibit phagocytosis, so you are seeing just the mechanism that is extracellular and is the net release. So if you incubate these neutrophils with the Shigella or Staph, you see a killing of the bacteria. But if you add DNAs to these cultures, you decrease significantly the death of the bacteria. And also, if you do the same kind of culture, instead of DNAs, you add an antibody that neutralizes the histones that are associated with this structure, you also decrease the killing of the bacteria. So this mechanism is something very interesting because it explained that at that time, something that was people knew and they didn't know exactly why. Some bacteria express DNases or nucleases. And now they realize that those bacteria are much more resistant to the NET-mediated killing because they produce their own DNAs to digest the DNA and then let the bacteria to escape. So this structure is microbicidal and has also the ability to trap and snare the different pathogens, trying to contain their dissemination in the body. And we start to look at this kind of mechanism using Leishmania. That is what I was working my whole life. And we demonstrate that the Leishmania amazonensis promastigotes that you can see here, beautiful with this interesting flagella, they induce the neutrophils to release this structure that's, again, stained with DNA, histone, and elastase. And here you see the colocalization of all those markers. And also, if you culture the parasites with neutrophils that we inhibit phagocytosis and we increase the capacity of these neutrophils to release these NETs with 4-ball myristate acetate, this PMA, that is a classical NET inducer. Instead of measure the Leishmania death, we measure the Leishmania survival because it's much easier. The parasite moves the flagella, so it's much easier to see. And comparing where you have a lot of death in the culture, because here you have the death of the parasite by the phagocytosis and by NETs. So if you inhibit the phagocytosis, treating the neutrophils with cytochalasin D, we increase the parasite survival, or we decrease the death of the parasite. And we found the same activity using the neutralizing anti-histone antibody. So without phagocytosis and if we neutralize the histones, we again, much more parasites are survived. And this is not the effect of the antibody because the isotype control was not doing the same thing. And we confirmed this kind of data showing in a live and dead assay that the parasites incubated with the NETs are dying, here stained in red, and some that are still alive are stained in green. And if we incubate these parasites with purified histones, we can see that some parasites are dead and some others are alive. So you can see here even a parasite moving very fast his flagella because it was still alive. So I hope this. If you, for you to have an idea how this mechanism happens, in this small movie, we are going to see neutrophils incubated with the parasites, that is this long parasites that are moving, in the presence of cytox green, that is a stain that intercalates in the DNAs when it's outside the cells. It's a vital dye. So you can see that as soon as the parasites are interacting with these neutrophils, some neutrophils, not all of them, start to liberate to the NETs, and these structures are fixing and holding the parasite. And more or less here, you're going to see a parasite that is trapped in this structure, and a neutrophil that's not releasing its nets. It's going to come here and phagocytose this parasite, you see. So more than only being microbicidal and trap, ensnare in the parasite, it also happens to help the phagocytosis of the different pathogens. So this was a review that we wrote in 2012 with a list of, at that time, the kind of microorganisms or molecules, cytokines, and microorganisms like LPS or nitric oxide and different molecules that were able to induce net release. And we add to this list the leishmania, because we show that promastigotes, emastigotes, and even the lipophosphogycan, that is a molecule that is in the parasite cell surface, were able to induce this kind of mechanism. And this is a list now that I don't know if it's showing completely, but we have now many viruses, because we just come out from the COVID pandemic. So we have different viruses that can induce the same this mechanism, including SARS-CoV-2, and also different protozoa, and many others. So it's a mechanism that is being related with different microorganisms and different stimuli that are able to induce neutrophils to release this kind of structure. In the lab, we are also collaborating with the group of Deborah Fogel, that is a lab involved in the study of amyloid disease and amyloid fiber proteins. So we described that 3 different amyloid proteins, transthyretin, alpha-synuclein, and a prion from yeast, the 3 of them induce net release from neutrophils, but not the soluble form of the protein or even the amorphous form of the protein. And here is a net induced by alpha-synuclein. And more interesting, in the field, there is a discussion about what is the toxic mechanism in this amyloid disease, like Parkinson, Alzheimer, and all that. If it's the fibers that are being accumulated in the nervous systems, or if it is the oligomers that are being produced by digesting this kind of structure. And what was very interesting is because we incubate the amyloid proteins that you see here in the picture of an amyloid protein. When we incubate it with the nets, they are being digested in oligomers. And if we inhibit, if you add it to this medium, an inhibitor of elastase, we inhibit this digestion. And more interesting is if we add this digested amyloid proteins in a cell and measure the survival of the cell, the amyloid protein was not killing the cell, the nets are not killing the cell, but if we add the protein that was being digested by the nets, we have a completely killing of the cells in the culture. So showing that really nets are able to digest the amyloid fibers and producing very toxic oligomers of these proteins. We also observe this kind of structure in the lung of patients with amyloidosis. So you see the DNA stone is an antibody that recognizes an epitope shared by DNA and histone. the elastase in this, and here is the colocalization of this tree staining, showing the whole structure of the nets. And also, we observed that we can find the amyloid deposit that is stained by Flavin S, that is a typical staining of these fibers, colocalized with the elastase, with the DNA in the skin of a patient with systemic amyloidosis. So we add another inducer in this list, that is the amyloid proteins. And more recently in my group, we find something very surprise for us, that is laminin, the extracellular matrix protein also can trigger the net releasing neutrophils. This was, in the beginning, we were interested in see what exactly mimetize more the site of the parasite during the inoculation by the sandfly. So leishmania is transmitted by the sandfly vector in a pool of blood, and where it meets the neutrophil for the first time. And all this is occurring in a extracellular matrix. So we want to see if we could mimetize this and see if the interaction of the neutrophil with the parasite in the presence of matrix proteins could modulate this kind of mechanism. And for our surprise, we first did a control, just incubating neutrophils with the laminins. And we, as you can see here, neutrophils are liberating nets. When we incubate them with laminin 111, 411, and 511, the three types of lectin that we tested. So we see the nets stained by DNA, by elastase, and also an antibody that recognizes each one of the forms. So some of the laminins are merged, are being stained close to the nets, like 111 and 511, and the other one just induced net and does not exactly colocalize. And here you see in different doses, we measure the DNA in the supernatant of the culture, showing that all of them are able to induce net release. But this was a surprise, and because we tested the laminins in solution, we said, well, perhaps we need to do something more close to the extracellular matrix milieu. So we polymerized the laminins with a treatment in acid pH. So you can see here that the polymerized laminin, the more interesting was the laminin 111, that it forms a matrix porous. So the net was here stained in green, the laminin is in red, and if you look at the neutrophils, we don't see them because they go inside and they are in the bottom of this thick layer. In the laminin 411, it's completely different polymer, much more thin, much more compact. And we see that even when they are polymerized, you see the neutrophils releasing nets stained here in green again. And here is the polymer of laminin 511. Again, you can see the nets and the neutrophils, the nuclei of the neutrophils in this structure. So it was not just the soluble molecule, also when they are polymerized, more close towards its appearance in the tissue, they all induce nets. So we added the laminins in the culture of neutrophil with Leishmania parasites. And as you can see here, all the three laminins that we tested, they increase the net release from that was already induced by the parasites. So all of them are increasing the amount of net of the parasite. So again, one more inducer of the net releasing neutrophils, and now protein that is in the extracellular matrix. This is not very uncommon because there are many different cytokines, for instance, that are able to induce net release, like IL-8, GM-CSF, and some others. So this mechanism was described initially in neutrophils, and most of the studies are being done in neutrophils. But there are different cells that are able to release its DNA to the extracellular medium. So here you can see that mast cells, they also are activating and they liberate nets that are holding parasites, bacteria here, and they also have some microbicidal activity. Eosinophils do the same. In this first work, they are showing that these nets are, the DNA from these nets are from the mitochondria of the eosinophil, but now there are some other works showing that eosinophils can also release DNA from the nuclei. Monocytes. So here is monocytes incubated with E. coli. So you can see the DNA is stained with intercalating dye, probably DAPI, and also with the antibody against histones. And if you increase the amount of bacteria in relation to the monocytes, you have an increase in the release of this structure to the medium. And more recently, even lymphocytes, they can release this kind of structure. So B lymphocytes stimulated with PMA, the 4-ball ester, they release DNA in the medium. Stimulated with anti-IgM and LPS or ionomycin, all of them are able to induce the release of these structures from lymphocytes. And this is really DNA because here is a CD4 T cell with the nets labeled with propylium iodide, if I'm not wrong. And if you add DNAs, you completely abolish the staining because the DNA is digested. And interesting, the group of Valderias Dutra, they show that CD8 T lymphocytes stimulated with anti-CD3 and CD28. They also release these structures to the medium, and even the Th17 cells, in this work, they generate some of this Th17 against a bacteria that is Cutibacterium acnes. And so they see that they could demonstrate that the Th17 cells release DNA to the extracellular medium when stimulated by PMA in bacteria or just by the bacteria. That is something interesting and intriguing, but anyhow, it was like that, and many works are showing that even lymphocytes, they can do the same mechanism. More than that, this mechanism is very evolutionarily conserved. So cat neutrophils, they release also this structure. Here is a work where the Toxoplasma gondii are being trapped by nets released from a cat neutrophil that you can see here in more detail. Dog neutrophils also, they release net when they interact with the Leishmania parasites. again, stained by DNA, by elastase and histone H2A, and here you have the colocalization of all these labels. Chicken heterophils that are cells similar to neutrophils in the birds, neutrophil-like cells from fish, from a crab. It's exactly the same kind of mechanism. And even oysters. Here you see an infection in the oyster, so there is some bacteria labeled in red, and the nets are labeled in blue. And here you can see a bacteria trapped in this structure. And this kind of mechanism in oyster is not related just to infection, like as we are showing here and here, but also to injury. If you do some injury in the oyster tissue, the cells can release their DNA, probably recognizing a dump that's being produced by the injury. This earthworm also, they release DNA from its cells, here labeled in red and the bacteria in green. The amoeba, one of the stages of Dictyostelium discoideum, the S cells, they also release this structure in the extracellular medium, and if you treat this with DNAs, you remove you digest the DNA, and so you don't see the stain anymore. And here we show that this mechanism also is occurring in the Periplaneta americana, that is this big cockroach, terrible one. So we see here the nets labeled with a stone in green and some bacteria that are being trapped. Here is stained in propidium iodide and in the scanning electron microscopy. And what is interesting, because insects normally they have as a defense mechanism when they are being invaded by a pathogen, something similar, the cells are agglutinated and then they are melanized. So because of that, you see this dark color. So we inject the body of the cockroach with the E. coli, and as you can see, you have this clump of cells, and they are being melanized exactly in the site where we inject the bacteria. But if you inject the bacteria together with DNAs, as you can see, you have different clumps, melanized clumps all over the body, proving that this structure is helping the cells to be together, to agglutinate, to form a clump, to contain the dissemination of the microorganisms that we inject here. And this is not an effect of the DNAs, because if you inject DNAs, you don't see melanization in the body of the cockroach. And the last example is plants. So if you cultivate plant roots with a fungi, this Nectia hematococca, you see that the roots are okay. But if you add to this same kind of culture DNAs, you start to see that the tip of the roots are being damaged. And this was, again, not an effect of DNAs, because DNAs alone was not doing anything in the roots. And if you look the tip of the roots more carefully, you can see that the roots, in the presence of the fungi, they are normal, they are okay, the fungi is here. But if you add DNAs, you have an increase in the fungal growth, and even the IFA are going inside, invading the tip of the root. So because of this, you are seeing this damage in the root of the plant. So it's a very conservative mechanism. I told you that the nets are a scaffold of DNA, of chromatin, associated with different molecules. So in the first proteomic study that they did in this structure, they found associated with this chromatin proteins from the granules of the neutrophils, from the nuclei, stones, and different other proteins, cytoplasmic solvent and glycolytic enzyme. As this study is being analyzed by other groups with different methodologies, you can see now that you have different proteins depending on the stimuli that you use to stimulate the neutrophils and the release of this structure. So here you see some nets produce number of proteins associated that are specific just for LPS-induced net or for IL-8-induced net, and some others that are shared. Here again, some other showing different proteins and ionophore, some specific proteins that associate just with the nets induced by PMA, and some others that are induced by another stimuli, and some proteins that are being shared. So what we know now is that depending on the stimuli, we don't know exactly how, but we have different proteins being associated with the chromatin of the neutrophils and being liberated in the extracellular medium. And our group was interested in see if this nets could also carry some microRNAs. So as you can see here, we found some microRNAs associated with the net structure, and we analyzed nets induced by the PMA, that's the classical net inducer, with leishmania and with transthyretin. And we could identify some microRNAs that are present in the nets induced by PMA and by transthyretin and not by leishmania. And here, another microRNA that is present in the net induced by PMA and by leishmania, not by the amyloid protein. And as these microRNA are not being released alone, normally they are associated with proteins. Sorry, this is just to show that the microRNA is present, is colocalizing with the net structure. But we also detected in this structure some proteins that are normally associated with the microRNA that are being released, like Argonaute-2 and apolipoprotein A1. So here we see that nets induced by PMA, they have the nets are stained by elastase here in red, and the Argonaute protein is stained in green. And also in the leishmania induced nets, we see the same thing. And also interesting, the leishmania is completely labeling with the antibody against Argonaute, something that we need to know exactly why. The same net induced by PMA, and here is stained by apolipoprotein, the one from PMA, and this one from leishmania. You see the nets, and they stain the colocalization of apolipoprotein in this structure. So in this work, we are detecting for the first time that not only you have not only proteins in the associated with the net scaffold, but you have also some microRNAs and proteins that are normally associated with these microRNAs. In the mechanism, how is the signaling, what we know about the signaling involved in this mechanism, the release of this structure. So in the beginning, it was shown that it was a completely different mechanism of death, because if you induce apoptosis in a neutrophil, treating the cell with an antibody against Fas protein, we induce apoptosis, and we can show that it was labeled, this cell was labeled by tunnel, and not neutrophils that are induced to necrosis with a Staphylococcus aureus pore protein toxin, and also the one that are induced to release nets with PMA. But if you look the supernatant of these cultures, we can detect the nets by staining with DNA. So we see very few with in the cells that are induced for apoptosis, for necrosis, but a lot in the supernatant of the cell that we induce net release with PMA. And actually, this is the assay that we use to study this kind of mechanism. You incubate neutrophils with the stimuli that you want to test, and then we measure DNA in the supernatant. That is the first sign that the cell is being released nets by this mechanism. It was also shown that elastase that are present in the azurophilic granules is a very important molecule for this mechanism. So what is being shown is that the activation of the NADPH oxidase with production and induction of the ROS production, it somehow changed the azurophilic granule, that is one type of granule of the neutrophils, and this finds the elastase to move out from the granule, go to the cytoplasm, and enter in the nuclei. And in the nuclei, it starts to digest the histones. This is important for the condensation of the chromatin. And after elastase goes to the nucleus, starts to digest the histones, myeloperoxidase, that's another enzyme present in the same granule, also do the same, also migrate to the nuclei, and by a different mechanism from the activation, the normal activation of MPO, it starts to help or increase the digestion mediated by neutrophil elastase. And in this picture, as you can see, you have a lung of a mice that was infected with Klebsiella pneumonia. And if you see that the parasite in this lung is completely covered, the parasite, no, the bacteria, is completely covered by net structure. But if you do the same in an animal that is elastase knockout, doesn't produce elastase at all, you don't see the same pattern. You have the bacteria in the lung, but you don't see the net structure in this covering and trapping this bacteria. So what we know now is that when the elastase goes out from the azurophilic granule until she arrives in the nuclei, it starts to digest the cytoskeleton of the cell. So elastase is a very important molecule. So it goes out from the nucleus, from the granule, enters the nucleus, and also digesting the cytoskeleton of the cells. And another characteristic of this mechanism that we can observe is the nuclear membrane. It starts to swell and disintegrate as soon as you have the chromatin being decondensed. And also the lamine layer in the nuclear membrane it starts to disassemble. We don't know exactly who is the responsible for disassembly, but anyhow, it is observed as the cell is entering in the NETosis. So myeloperoxidase is also an important enzyme, as I mentioned earlier, but for some stimuli and not for others. So if we incubate the neutrophils with PMA, and we have some neutrophils that are myeloperoxidase negative, they are deficient in this enzyme. As you can see here is a normal one, is stimulated with PMA and releasing the NETs, and the other one that is deficient in myeloperoxidase doesn't release NETs at all. And it's exactly what we are seeing here in this graphic, that we can obtain also in incubating these neutrophils with myeloperoxidase inhibitor. But this is not true, for instance, for Pseudomonas aeruginosa, because the MPO deficient neutrophil, they still, in these white columns, they still release NETs. And even if you try to inhibit the enzyme, you don't see any effect. So depending on the stimuli, myeloperoxidase is important or not. Also, there is another enzyme, the peptidyl arginine deaminase, that we call PED4, is another important enzyme in the decondensation of the chromatin, because it citrullinates the arginine in the stones. So changing the stones composition and changing the behavior of these stones, it starts to contribute for the chromatin decondensation. So here we have neutrophils from a PAD4 knockout animal, so it doesn't release NET in contact with E. coli, as the neutrophils from a wild type animal that we see this beautiful NET being released. And also, we can detect the citrullinated stones in the stones present in the NET structure. Here, stimulated by a calcium ionophore. It's also very important for most part of the inducers, the activation of NADPH and ROS production. So here you see the NET DNA being released to the extracellular medium. And if you incubate this neutrophils with NADPH oxidase inhibitor, like DPI, you don't see this release. And this mechanism is confirmed in neutrophils from patients with chronic granulomatous disease, CGD patient, that they have defects in the NADPH oxidase, so they don't produce ROS. And so if you incubate here neutrophils with the Staphylococcus aureus, that's stained in blue, they phagocytose the bacteria, but they are not releasing NETs. Remember, this is a bacteria that is first demonstrate the NETs in the paper from Zipinski group. And also, if you treat these neutrophils from CGD patients with PMA, you don't see NETs at all. But if you add another an extracellular source of ROS, then these neutrophils release their NET. So it's an important mechanism for most of the stimuli. And now something interesting, because it was shown that elastase, digesting the cytoskeleton, going to the nucleus and digesting cutting the histones, it also can cleave the gasdermin D in a fragment that is active to make pores in the membrane. And so, in the way that elastase goes from the azurophilic granules to the nuclei, it can cleave the gasdermin D in a fragment that is potentiating the effect and is disturbing not only the granules membrane, but also the nuclei membrane. So it's being cutted and being inserted in the plasma membrane and in the membrane from the nuclei and from the granules. So increasing the release of elastase and making easy the elastase go inside the nuclei. So it has been shown that gasdermin D is not only cleaved by caspase 11 from the inflammasome, but also from the serine protease from the in the neutrophil. So this is a big issue now, because there are some people that saying that even caspase 11 can go into the nuclei and also digest or participate in this kind of process. So caspase 11 not only is going and doing the activation of cytokines, but also migrating to the nuclei and contributing for the condensation of chromatin. And this paper I brought because of the talk of Igor yesterday is a group from Denisa Wagner, and she's showing something very interesting, at least for me. When you stimulate neutrophils with nigericin and also with ionomycin, you see the nets, but also you can see the speck formation. And even sometimes the speck formation is colocalizing with the net structure. And here you see that animals that don't produce the peptidyl-arginine-deaminase, the PAD4, in the myeloid cells, they don't form specks. So there is some talk between the NETosis, perhaps, and the inflammasome activation. So you see this for the ionomycin and also for nigericin. And interesting, if you inhibit NLRP3 with this compound, we don't see the nets in the induction in the neutrophils, even when they are stimulated with nigericin or ionomycin. So you see the nets here, in this shown by this red arrows, and here you don't see the same structure. So perhaps there is some talk between these two pathways. And here is just the quantification of this figure. So what they are saying, they are showing in this paper is that PAD4 somehow is increasing the amount of NLP3 and ASC. And this is activating, of course, caspase 1, that's cleave gasdermin D, and this is making pores in the nuclei membrane and also in the plasma membrane, and this is helping the release of the nets to the extracellular media. So this was a discussion of a group of people that are working with nets, and so they did this interesting chart, because there are some findings in the net mechanism that are confirmed and everybody agrees, but there are many others that people are still discussing if it's participating or not, or for some stimuli, not for others. So there are still a lot of discussion about this mechanism of release of net to the medium. More than that, it was described in 2010 that neutrophils can release the nets not only by this mechanism that I explained until now, that is being released with the neutrophil death, but also there is a mechanism that they call early and non-lytic net formation. And they have nice results showing that the neutrophils is releasing the DNA by nuclear vesicles. So the DNA is being decondensed with the PAD4 activation, like the lytic mechanism. And so some vesicles are taking this decondensed chromatin, and this is being released in the extracellular medium, and probably is being associated with proteins from the granules that are being released at the same time. So this is a non-lytic mechanism described by the group of Paul Cubes from Canada. And they have a very beautiful work showing by in vivo image that when they infect mice with Staphylococcus aureus, they can see in these mice neutrophils, normal neutrophils with the nuclei and the cytoplasm, the normal neutrophils. They observe some neutrophils with diffuse nuclei and some other ones without nuclei, like this one, just the cytoplasm. And more interesting, this mechanism of release of net without the death of the neutrophils, those ones, they can migrate, they are chemotaxic responding, and they even can phagocytose the bacteria. So it's some other mechanism that we need to pay attention because it happens much quickly than the other one. In 10 minutes, 15 minutes, we already can see the DNA in the supernatant. And we don't know yet if this is a beginning of the mechanism that is going to then be in the lies, or if it is something completely different. We have shown that leishmania induce both mechanisms. So if you incubate neutrophils with the parasite, 10 minutes later, you already can see the DNA in the supernatant of the culture, and it is elastase dependent, but we could not observe ROS production. That is normally what occurs in this other mechanism, but we also didn't see any PAD4 dependence, different from the classical NETosis, that's the lytic one, that we have participation of ROS production, elastase, PAD4, but we don't see MPO dependence in this process. And looking the signaling pathways induced by leishmania in neutrophil, we could observe a pathway that is dependent of ROS production, and another one that is more calcium dependent. So we need to know now which one of these pathways is responsible for the early NETosis and for the classical or lytic pathway. So, but this is the effect of nets in parasites, so how it's happened, and this structure is being released in the extracellular medium, so they can modulate different cell features of the cells that are in the same milieu. So they can interact with different cells in the milieu where this structure is being released. And we touched this a little bit because we are trying to see the monocyte differentiation in dendritic cells by incubating these cells with IL-4 and GM-CSF. And so if you do that, the cells is decreasing the expression of CD14 and increasing the expression of CD1A. But if you do this in the presence of NETs, you can see that not all the cells are losing the CD14 expression, and also not all the cells are expressing CD1A. This is the percentage of cells, and this is the MFI. So you have the same kind of results. And here, just a picture showing that we are incubating the cells with the whole net. It's not a fragmented net. And what we discover is that the nets are digesting the alpha chain of the IL-4 receptor, but not the GM-CSF receptor. And here it's just for a quantification of this digestion of the IL-4 receptor in the cells treated with NETs. And if you add elastase inhibitor, you restore the expression like in the control. And also if you digest if you degrade the NETs, is the same thing. And if you incubate the parasites with this kind of cells that are being differentiated in the presence of NETs, you can see that you have an increase in the parasite multiplication inside of the cells. So what we are saying is that NETs produced in the milieu where the monocytes are being differentiating in the dendritic, possible dendritic cells, they are decreasing the IL-4 receptor. I didn't show, but we have these results. They are increasing CD68, CD14. They produce more IL-10 and TGF-beta. And so all this mechanism is more like an M2 macrophage that are allowing the parasite survive. On the contrary, if this same differentiation process is occurring without the presence of NETs, you have dendritic cells with all the characteristic markers producing IL-12 and TNF-alpha, so you have more contributing more to the parasite death. We also did that with HIV-infected macrophage. So here is just for you to see the macrophage culture incubated with the NETs. Again, well, it's not net digested, it's completely structured. And when we incubate the neutrophils, this is the macrophage from normal people that we infect in the lab with HIV. And as they are incubated with the nets for 3 hours, then the cultures are washed and we put the HIV, and we look this in different concentrations of the nets added to this culture, and you see a decrease in the HIV-1 replication. And this happened with nets from normal neutrophils, and neutrophils from normal donors, and also with neutrophils from patients infected with HIV. Interesting. If we recover the HIV from these cultures and we try to infect other cultures, they show less infectivity. So there is something happened in this HIV that are being replicating the neutrophil in the macrophage, in the macrophage that were treated with nets that are making them less effective. And we are trying to see exactly what's going on. What we know already is that this macrophage treated with nets, they produce hunches and MIP1 beta that are molecules that are inhibiting the co-receptor of HIV. And also we detect nets in the plasma of the patients infected with HIV-2. So everybody was very happy because, well, we have another mechanism that is able to contain the dissemination of microorganisms, and they can protect the host from the different kind of pathogens because they are also toxic for many pathogens. But we discovered that there is also association, a strong association between nets and some disease. So there is the physiological and the good side of the net mechanism that is trapping and killing microbes and contributing for the clearance of this parasite and the net structure, and is an effective mechanism. But we have also some way when you have a increase in net release or in a defective of the clearance of this structure. So you have this structure being in contact with the immune system. So you have autoantibodies, and this is very nicely shown in the lupus erythematosus systemic, because you have the anti-nuclear antibodies, and you see the nets being released spontaneous by these neutrophils from these patients. We have the release of many dumps and allergens that are causing disease, and also nets are very important in the coagulation system. So we have many problems in thrombosis with this structure. So in the infection mechanism, if you don't produce nets, is bad because the parasite is taking advantage of that. But if you, on the other way, if you produce too much, or if they are not being cleared, so you also could have some problems because of the potency of many molecules in all this structure that is being released. So depending on the organ that this kind of structure are being produced, we have already many diseases related, like Alzheimer, stroke, gout, different diseases. They even are involved in the gallbladder, gallstone, and more recently, in a nice paper that are from the Fernando Cunha group, they show that in patients with COVID-19, you have a lot of nets in the plasma, in the tracheal aspirate, in the autopsy of the lung. Oops. And more than that, these structures are very toxic to lung cell A549. So they show this by labeling the cell with cytokeratin 17, that is normal staining. And so if you incubate the cells with NET, you see that you have a toxic effect in these cells, and this probably is contributing to the COVID pathogens, not only damaging the cells in the lungs, but also is related. There are many works relating the net release with thrombosis, because it's a very strong activator of the coagulation. And also there is a lot of work trying to inhibit this kind of mechanism. So trying to use TLR inhibitors, because they are a potent inducer, inhibitors of other molecules related with this structure, a Ross scavenger, peptidyl-arginine deaminase inhibitors, and DNA therapy that is already being used in cystic fibrosis, that you have a lot of NETs in the contributing to this disease. And I heard that there is also some people trying to do some clinical studies using PARP inhibitors for treating COVID-19. We saw that you have NETs in the leishmaniasis lesions, and it's in some patients, you see this huge amount of NETs that are here stained with peroxidase, and you see even the parasites in the tissue associated with this structure, not only by this kind of microscopy, but also by fluorescence. You see the mastigote nest associated with NET stained with histone, antibody against histone and anti-elastase. So you see a very close proximity of these two labelings. And I finish thanking all the people that did this work with me. I'm saying the name of the students in the works, but I'm collaborating a lot with the lab of Deborah Fogel and Stefania. Tatiana is the one that is an expert in the laminins in polymerization and all that. Marcus and Luis is the ones that are involved in some of the signaling pathways. Lucia is the one that is helping with cat and dogs NET experiments. Domicile group with Andreas and Jairo in HIV, Ingo and Marcelo and Savino in the work with laminin, Fernanda and Fatima in the leishmania together with Rodi, and also in the dog experiments, the group of UFOP, Rodolfo, Alexandre, Kelvis. And of course, I need to thank the financial agents, FAPERG, CAPES, and CNPq, and the blood that we get from the Hospital Universitário Clementino Fraga Filho. And thank you very much for the invitation again and for listening, and I'm ready for questions.
Thanks, Elvira. It was amazing.
Excellent.
At the same time,
Amazing.
Really a broader view of NETosis, many aspects, and deep in every single aspect. So it was really a masterclass. Really cool. So we have a first question from Andrew. Do you want to start, Andrew?
Yeah, sure. Hey, Professor, nice presentation. I really enjoyed to see new papers from the group. And my question is related to the capacity of NETs to modulate the immune system. Do you believe that NETs could improve or decrease the immune response to a vector vaccine strategy? Because I work with that, and I will also work with Gasdermin T knockout mice. So I am very interested in this part.
But what do you mean? If the vaccine could activate this kind of mechanism?
Sorry,
Or
I
if
cannot
the
understand you.
You are asking if the nets are being could help the mechanism, or if they are affecting the mechanism, or if the vaccine is inducing nets.
Yes, exactly that. If they are affecting the mechanism to the immune
Response.
system to respond the vaccine
Mhm.
or improve.
Well, it depends on the kind of vaccine that you are talking about. Probably
I work with a bacterial strategy vaccine.
So if you are using the bacteria, of course, it can induce net release. And there are some groups that are also trying to use the net as an adjuvant. But then you need to inactivate the enzymes, because you have a lot of different proteases in the structure. So we need to inactivate this, and so they are trying to use this as an adjuvant. But you need to try to see if your vaccine, if you are using the whole bacteria or a product of the bacteria, if they can interact with neutrophils and if they can induce net release. There are a lot of toll-like ligands that are inducing net release. So it's something that you need to try.
Okay, thank you very much.
Thank you for the question, Andrew.
Thank you.
Nice to see you again.
It's nice to see you too.
Natalia, do you want to follow?
Yes. So thanks for the presentation, teacher. But my question is not so. Actually, my question is more easy than Andrew's question, because when you were talking in the beginning, and if I could understand correctly, neutrophils are present whenever an infection occurs, right?
Right.
And you commented that the neutrophil can release its content like DNA or stones to extracellular medium to form the nets that will cause the microorganism to be trapped. So I was thinking about phagocytosis, because when we learn in the school, or even if in the undergrad program, we learn that the neutrophils like do the Pac-Man. They just eat like that. And then I was thinking, when you are talking about that, they release the content, the way that we learned about phagocytosis from neutrophils, they still exists, or all neutrophils release their nets to trap the microorganism?
These are two different mechanisms. You have some neutrophils that still are phagocytosing, at least when you incubate them with Leishmania, for instance. We see some neutrophils that are phagocytosing the bacteria and are not releasing nets, and some other that are just releasing nets. We don't know, we don't have markers of subpopulations of neutrophils, so we don't know if it's a different population of neutrophils that are more phagocytose prone and the other ones that are more net prone. We don't know if it's a kind of age, so neutrophils that are already almost dying because they are too old, they don't live a lot, so these ones are more prone to release nets. So this is a mechanism that we don't know yet, because we don't have markers to separate neutrophils so easily as you can see for lymphocytes, for instance. And there are some very few works that they have some data that the neutrophils sometimes induce, depending on the microorganism, phagocytose the microorganisms and then release nets. But these are few ones, and we don't know exactly why some do something and the other thing. So we don't know yet. But both mechanisms occur. what you learn in your graduation are still happening. The neutrophils are very active in phagocytosis.
Yes, I was asking because I'm not from the area. I do not work with lysis or not at all. But and then I was like, actually, you just answered my question, my second question, that we can't separate those who are just releasing nets and those who are just phagocytosing the I don't know, the bacteria or something.
Yeah, no, because we don't have markers for subpopulations of neutrophils. The only thing that that you know is that you have some neutrophils that instead of when you obtain the neutrophils in the gradient, normally the normal neutrophils are in above the red blood cells layer, but there are some that are has a lower density that start they in the gradient they stay exactly in the same place as the peripheral blood mononuclear cells, and so those neutrophils they release more. They are more prone to release nets than the normal ones. They release much more, and they have different densities in the gradient, but it's a different kind of neutrophils, not the normal ones that you have the majority in the blood. So it's the only thing that we know. At least in lupus, for instance, we don't know exactly why these neutrophils that are different from the normal ones, the low density, they are releasing a lot of nets. We don't know exactly which is the inducer, but it's something that they found in the paper, in the people that is developing this kind of disease. So those neutrophils are releasing more nets. It's the only thing that we know different.
Okay, thank you.
Karina, can I skip you for a second and ask Renata to go for the next question, please?
For sure, for sure.
Thank you.
Tem certeza? Okay. My question was related to Karina, Natalia, I'm sorry, Natalia's question. I was asking myself, I saw in the time-lapse images that you show in the presentation, that some neutrophils phagocytose, some release the net. So I was asking myself if there is such a negative retroregulation that regulates the net, the non-releasing of the net in those neutrophils that only phagocytate.
Yeah, it's something that we don't know yet. We don't know why some neutrophils release nets and some others phagocytose, some others do degranulate. It's something that is not known at the moment. We just, if you look if you purify neutrophils like I show in the video, and you put them with any kind of stimuli, you are going to see that some are releasing and the other ones are moving around of those structures, phagocytosing what is trapped, and some others is releasing nets. So it's something that we observe, but we don't know exactly why they do that. And mainly because we don't have markers to see if there is some subpopulations of neutrophils, some ones that are more prone to release nets and some others that are more prone to phagocytose. So we don't have markers to separate the cells, so we don't know exactly why one of these neutrophils they release net and the other one phagocytose a microorganism, for instance. It's something that we
Did you try use the supernatant of the cells of the neutrophils that release the net to see if they can inhibit neutrophil net release?
There is one, we tried to do this, but it was very difficult at the time that we planned to do that. So didn't reach any conclusion. But there is one work that they did that, and actually if you incubate neutrophils with nets made from other neutrophils, you increase the amount of nets that you recover in the supernatant. And I was going to say something more. I don't know. I forgot. If I remember, I tell you later. Sorry, I remember. Nets are very toxic to different kind of cells. For instance, to epithelial cells, to endothelial cells, to the lung cells, as the group of Fernando Cunha showed. But as you can see, neutrophils, at least some neutrophils, they are resistant to the net-mediated toxicity, because you see the net being released and some other neutrophils going around and doing what they want to do without any problem. So it's something strange.
Okay, thank you.
Also are resistant to nets, because when we incubate the macrophage with nets, they are fine. So they endocytose this structure and they are not being killed by the nets.
Do you know what? I'm sorry, Professor.
No, go ahead.
Wanna comment, Gustavo?
Do you know what gives these cells immunity or resistance to cytotoxicity?
No, we don't know. We don't know exactly. I'm saying they are not toxic. For instance, we show that nets are toxic for leishmania, for instance, for promastigotes. But mastigotes, we tested, and they are fine with this structure. They are not killed by the nets. But we don't know exactly what's the mechanism. Of course, this is a parasite. And macrophage, we did experiments with the ones that I show incubating monocytes, and also the ones with HIV that incubate them with the macrophage. At least in the conditions that we didn't observe any toxicity for these two types of cells. So
it's interesting.
We don't know exactly what makes them resistant, but they are not dying.
Wow, thank you.
You're welcome. Thank you for the question.
Gustavo, you wanna comment?
Yeah, no, I was just asking because during your answer, you mentioned the age of the neutrophil, and then this is kind of a simple experiment to collect neutrophils, weight, different time points, and then try to induce or give bacteria or leishmania. So have you done that and see whether with time they will be more frequency of net producing neutrophils increase compared to early time points or things like that?
Yeah, we never did that, because if you purify neutrophils and you put them you wait them in the medium, is what you are saying. You purify neutrophils and then you test 1 hour, 5 hours later, then they start to go in apoptosis too. Because neutrophils are, even when we purify them from blood, we need to do this at the same day that we collect the blood. We already try to take the blood, the whole blood that you got from the blood bank, to purify next day. So they are not the same. It's a very complicated cell to
yeah, I know it's a very short-lived, but maybe in a smaller, like shorter time course, you could at least try to address whether, by
it's something that we could do and see what's happened. There is some works where they test neutrophils from children and compare with adults, and some people find difference, some others didn't. So it's still not clear. In the beginning, it was something very attractive to say, wow, they get old and so they die in a way that they are sendo benéfico to other cells, but this was not confirmed by different studies. Some found this, some others no.
Yeah, but a neutrophil from an old person and a neutrophil from a young person is, the age is similar, okay, of the neutrophil itself. The age of the person is different, but the neutrophil borns every 6 hours. Okay?
Yeah, but
I would think that if you know, neutrophils, as you said, neutrophils are towards the 6 hours, they will be more prone maybe to something. If it's just a freshly born, then yeah, then maybe, I don't know if there are people with setup to differentiate neutrophils from like a stem cell like or something like that, because then you could synchronize and then try to do a little things like that.
Yeah, I don't know if there is someone doing this with neutrophils. I mean, differentiate them from them. But if you test mice neutrophils, if you take my neutrophils from blood, bone marrow, and the problem is because when you remove them from the peritoneal cavity, you normally prime them with something. So, but there is no much difference. And also, it's difficult to compare the different studies because each group is using a different kind of stimuli. And as you can see, there are some stimuli that do something completely different from the other ones. And now, what I was more interested is in this talk between the different pathways, because with this paper from the Denise Wagner group is very interesting because you have the inflammasome activation, at least with these two kind of stimuli that she used. So you have NETs and you have the SPEC formation. It's something interesting. And also you have the PAD4 affecting the ASC and NLRP3. So it's
maybe it is related to the cytoskeletal, something related to the cleavage of some proteins from the cytoskeletal.
Yeah, but of course you have this all this. I imagine the whole DNA being decondensed and pressuring the membrane that is already with some pores, with the Gaussian pores. So this is, everything is going extracellularly. But you have the SPEC formation. The SPEC is going together, at least what they show in this paper that I just mentioned. It goes together, but it was activated before.
Yes, but when, if I'm not wrong, I understood that if you inhibit DNase, you reduce the ASC formation. So it could be related to this cleavage in the cytoskeleton proteins, maybe, no?
Yeah, they didn't inhibit elastase. They inhibit
DNase.
No, they don't inhibit DNase. They inhibit the NLP3, and the DNase is working after the NET is being released, not before. Just when it is outside in the milieu, then the DNase is working. They inhibit PAD4. They inhibit because they use a knockout, so they inhibit PAD4 and
they inhibit PAD4. Yes,
yeah.
yes.
And they show that PAD4 is increasing ASC and NLP3.
Yes. That's
I
true.
was even wondering if this could be one of the molecules that are being interacting with the inflammasome that Igor talked
Maybe.
yesterday. Yeah. So.
All right, let's move on with Hugo's question now, please, Hugo.
All right. Can you hear me?
Yep.
Sure.
Okay. Hi, Professor. Thank you for a great representation. I'm sorry if it is a silly question because I had problems with my internet connection and I lost some pieces of your presentation. But I was thinking if there is a possibility, in your opinion, or if there is this information in the literature, the modulation, how can I say that? If the nets provokes nets could modulate the monosystem and increase the tolerance, for example. I know the nets in general is associated with the pro-inflammatory response. However, in the content of the net, and when there is this release, if it's possible, increase the tolerance or cause other kind of effects like I'm not sure, anti-inflammatory, other effects not involved with pro-inflammatory response. There is something in literature, or do you think it's possible?
There are some data in the literature showing that nets could be inflammatory or anti-inflammatory, because there are some papers showing that when the net is released, because all the proteins that are associated with this structure, they can digest cytokines, for instance, pro-inflammatory cytokines, so they decrease the inflammation. But I don't know any work about tolerance, net tolerance. And I don't think that it's going to generate tolerance, because it's something that is contained inside the cell. As soon as this bunch of molecules is liberated, if they are not cleared very quickly, they start to stimulate the immune system. So you have antibodies against different molecules that are present in the nets. And sometimes these antibodies are even provoking diseases. So I don't know any work about net tolerance. And I doubt that it's going to induce tolerance. I know that they can be, depending on the context that it's being released, they can be anti-inflammatory, mainly because of this digestion of the cytokines that are being released by the proteins that are associated with the nets.
All right. Thank you so much, teacher.
You're welcome.
Lucas, you're next.
So can you hear me?
Yes.
Okay. First of all, thank you, Professor, for your great presentation as well. I'm also afraid it might be a silly question.
There is no silly question. There is no silly question. All the questions are interesting. There is no silly question.
So I've also had some technical problems, so I couldn't understand everything you said during the presentation. So perhaps you've even mentioned it, and it is related to what was already asked. Could that be a difference? I mean, is the stimulus is the stimulus related to the intensity of NETosis? Is there possibility for even the difference between a NETosis type neutrophil and a phagocyte neutrophil be related to the stimulus?
I don't think so, because as I mentioned, and I hope you could see the movie that I showed. We have in the same population of neutrophils, with the same amount of parasites. Some are phagocytosing the parasite and some other ones are releasing nets. So I don't think that is difference in the amount of stimulus, if it's what you are asking.
Yes, but I mean, like, if the intensity of these stimulus are different, like if you were to do a test with different, for example, if you, I don't know if LPS could trigger it, and
Yes.
if you would test it, yes. So I saw it on your presentation, so I wasn't afraid to mention it. But if you were to do a test with different concentrations of LPS and see if the intensity of this stimulus could influence the subset of each neutrophil.
And normally when we are studying the net release, we are looking the DNA being expelled from the cell. And when we look at that, if you put more stimuli, if you increase the LPS concentration, you get more nets in the supernatant, for instance. You see more nets stained by monofluorescence, for instance. But we are not looking the phagocytosis. And all the people that, there is a paper from the Ziklinski group that they show that, I forgot I think that was elastase. There is a difference in phagocytosis. The phagosome is going to fuse with the different granules from the neutrophils. So you have elastase and myeloperoxidase in the vacuole. This internet connection is mine or is yours? Are you hearing me?
Yeah.
Yes, I can hear you.
Oh, okay.
Perfectly. Yeah.
Just appear a message that the internet was not very okay here.
Oh, probably
so
it's
you
my
have
connection is poor.
Welcome to the federal universities.
Yes,
well, I am in a federal university, so I have the same problem.
Yeah.
So then you see the phagolysosome fusing with the granules of the neutrophils. And so perhaps it's the phagocytosis that is the trigger for the granules fused with the phagosome. And but this is not possible to test with LPS as molecule, you need to test with bacteria. So even though we are going to see some neutrophils that are phagocytosing and some other ones that are not. So it's
yeah,
I think that we really need to have a way to study subpopulations of neutrophils. There is already N1 and N2 neutrophils in cancer. It's already shown, but it's not a membrane marker. It's just some products that are being released by neutrophils that have an N1 phenotype and the other ones that are N2 phenotypes. So there is no labeling that you can use to separate the cells. So your question is interesting, but we don't have answer at the moment.
Perhaps the chemical nature of these stimulus could be something to look at.
Yeah, well. But you see that even the kind of stimuli, we are using the same stimuli for parasites, for fungi. It's the same one. One part of the neutrophils are phagocytosing, the other ones are releasing nets, and it's the same composition of the surface of the microorganisms that is easier to see a phagocytosis. So it's exactly the same in the population of the neutrophil that you purify. Some are doing one thing and some others are doing something different.
it's always the same with all the non-triggers.
I couldn't hear what you are saying because it was.
Oh, okay. My internet connection is poor, but no, it's all right. And no, it's all right.
If you write in the chat, I can. It's because it's picotando.
It's not
I
really
don't know
good.
how to say picotando in English.
Hiccups. I'm having technical hiccups.
Yeah, no problem.
But no, it's just like I don't know if perhaps if you had like different chemical different stimuli from different chemical nature, for example, LPS and different parasites and so different triggers. And if you've seen that, it's the results are always the same. So regardless of the chemical nature.
Okay, but there are some stimuli that induce NETosis and some others that don't. For instance, Zika virus, it does induce net release. It was shown in a nice paper by Fernando Cunha's group 2. So Zika doesn't induce net release. Chikungunya induce beautifully. So it's different, but then one induce and the other one doesn't. But they didn't look if the Zika virus are being phagocytosed, endocytosed, or phagocytosed. So it depends also. There are some stimuli that induce and some others that don't. But the ones that induce, they induce release of net in one part of the population and not in the other.
Carol, do you wanna?
Thank you very much.
Ask your question. Sorry, Lucas. Carol?
Hi, you're listening?
Yes.
Thank you for your presentation. That's very new to me, so I'm very excited and I love it. So I'm asking an actual curiosity. You speak about nets in autoimmune disease and also in the previous questions from Lucas about tumor-associated neutrophils. And I was wondering if and how nets can help or not help tumor cells to the progression of tumor, and also in metastasis. Maybe there's is any related to nets?
Yes, there is a lot of papers looking nets in cancer, in different kinds of cancer, and nets are related with metastasis, is increasing metastasis. Yeah, and there are some, well, I collaborate with a group of cancer here, and we show that vesicles, extracellular vesicles released from breast cancer, they induce net release. So there are some, at least extracellular vesicles, they induce net release. I don't know exactly the whole cancer cell, but probably. And they are very much related with cancer and also with metastasis. There are some papers already. Yeah.
It's not anti-tumor, it's pro-tumor.
Yeah, it's pro-tumor.
Oh, that's amazing.
Yeah.
Thank you.
So, Michelle?
Thank you, Professor, for the amazing presentation. You mentioned that some neutrophils no longer present a nucleus, and that's very amazing to me. And I wonder what kind of stimuli triggers this kind of net release, and how these cells are still viable and active if they don't have a nucleus anymore.
I couldn't hear the whole.
I think there's a problem.
Are you there, Elvira? I think she just froze. Let's wait a little bit.
I think that he's freezing.
Oh, she's coming back.
Elvira? Okay, now.
Do you hear
I
the
think
question,
now we are all here.
Ricardo? Or- I couldn't hear, just,
Michelle is gonna try again. Let's see.
okay.
Okay. You mentioned about that some neutrophils no longer present a nucleus, and I wonder
No longer
what kind of
presented?
stimuli.
Michelle, no
nucleus.
longer presented nucleus. Ah, okay. Well,
Yeah.
this was shown with mice injected with Staphylococcus aureus, and it was in vivo image. And if you want, I can send you the paper. It's by Paul Cubes. It's very nice. They have incredible movies. I'd like to have the same kind of apparatus to do the same here. We are seeing that in the mice ear injected with leishmania. That is beautiful. You have the neutrophils coming, and you have them releasing that. This is nice. But he does in the mice injected, and he does also in liver with other stimuli. But this one that I showed was mice injected with Staphylococcus aureus. So he was seeing the neutrophils moving, and some neutrophils without nuclei with an Staphylo inside. And they measured the chemotaxis of the neutrophils without nuclei. They measure the ones that are still with this diffuse nucleus. And so they move differently from the ones that are with nuclei, but they still move. They still respond to a chemotaxic cytokine, for instance.
That's nice, and that's very interesting to me. Thank you.
Yeah, I was in a meeting where Paul Cubes are presenting some of his research, and he presented exactly this kind of thing, and everybody was saying, "Wow, how could the cell?" We don't know how long this cell is living. If it's rest for long or if it's going to die quickly, we don't know that, and he doesn't too. But he was mentioned some old work. He couldn't say the author, but he was saying that there is a technique that you can take the neutrophils and centrifuge them in some speed that he didn't mention. So you remove the nuclei of the neutrophils. It's a technique that you can remove in vitro, the nuclei from the neutrophils. And even these cells without nuclei, I don't know how they survive, but he was mentioned that they can move and phagocytose without nuclei. So it's something amazing for me.
Elvira, I worked with that many times. So this is called cytoplasts. You have to treat cells with cytochalasin B. And then by differential centrifugation, the nuclei extrude from the cell, and then the membrane recompose. So the cells become metabolic active for a while. And we have a paper showing that cytoplasts undergo apoptosis by different stimuli as much as the whole cells. And that was the time that people did not know the composition of the pathways and whether, some sort of cancers, apoptosis, they were claiming that apoptosis needed the nuclei event. So it was quite nice. So yeah, the cell it remains metabolic active for a while. Then they die because, of course, not all the pathways survive. Then they lose.
But this is interesting. I'm going to try to find this and see if I can study some mechanisms of this kind of cytoplasm.
I think I have protocols for getting cytoplasm.
Great.
I can send you.
Yeah, but this is amazing because you are even putting cytochalasin, so you are changing the cytoskeleton with actin. So how it's unbelievable. He showed a movie of this kind of stuff, but wow.
Yeah, it works.
So it's not
I can tell that it works.
yeah, great. So I'll try
send
to
you the
see.
paper, our paper as well.
Please do it.
Renata, do you have a question before I give
Yeah,
it
me
back to
again.
Karina?
Just one. I was thinking you told about the conserved mechanism, that is NETosis, and you talked about amoeba. They are present in amoeba. There is it's a little weird, don't you think? There is present in amoeba because the amoeba can get away from the pathogen, first of all, and second, once the amoeba release its DNA content, it would die. So it's not very effective mechanism of defense in this case.
Well, I'm not an expert in amoeba.
No, I was curious.
But let me tell you, it's not the amoeba, well, at least not the amoeba that I'm thinking about, the big one that endocytoses a lot of things and all that. Is on the stage of this free-living amoeba. It's the cell that they call S. It's not the amoeba, the one that you have in the culture, this one that is eating everything that you give to them. Is on the stage of the amoeba that is called this S cell, and is a stage that is during the time that amoeba is free in the environment. Is one of the cells of the stage of the amoeba, free living amoeba. I'm just looking.
I was curious. I was very curious.
Well, I don't know exactly how the cycle of the amoeba. I just know that one of the cells during the cycle of the amoeba, it's doing this, and exactly how, I didn't follow if they publish another paper about that. I don't know if they look what is the meaning of this, but perhaps it's because it is in the environment, so it needs to, it's not phagocytosing. This is completely speculation. I'm not an amoeba person. So perhaps it is being attacked by some microorganism in the environment. So I really don't know. They just show that this stage they release, but I'm not following what's going on if they pursue this later.
I'm gonna search
something.
No, it's okay. Thank you.
Okay, Karina, finally you. Thanks for waiting.
My gosh, I think I forgot my question. No, I'm kidding. So my question is regarding in regards to the negative regulation of NETs, because you went deeply on the molecular regulation of this kind of depth and the effector mechanism, especially those related to the activation of these pathways. But I don't know if I missed something, but I don't see much about the negative regulation. So could, for example, autophagy be involved in this negative regulation of NETosis and so on?
There is a paper correlating autophagy with NET release. They couldn't find NET in a ATG5 knockout mice.
Maybe, I imagine.
So this is something that I'm almost sure that all those mechanisms, they have a talk together inside the cells. So there is this one with autophagy, now it appears this one with inflammasome. And I think that probably there are some other mechanisms that are related to this.
Because I started thinking about the requirement of acid pH of the cell for these enzymes work. And I started thinking about the possibility of baphyllomycin or some kind of these treatments for, I don't know, therapeutic intervention, something. And then I remembered the autophagy, because autophagy is involved in this cross-regulation of all or almost all of these inflammatory cell death modalities. And in regards to this pH requirement, acidic pH requirement, do you comment?
I don't remember if I'm not wrong, is acid pH is the one that is increasing the net release. I'm almost sure that is acid.
Yes, maybe, yeah.
And if you look some EMs, electron microscopy from the neutrophil, sometimes you see the autophagy structure to get inside the cells. And there is this paper that I forgot the authors, but they are showing some correlation between autophagy and NETosis too.
So beautiful, amazing.
Yeah. And what they are trying, at least in this patent site that you look, they are trying to test PAD4 inhibition, but not all the stimuli are by PAD4, but at least some. COVID-19, for instance, SARS-CoV is PAD dependent, and also after the net release to treat it with DNAs. So there is DNAs for cystic fibrosis, for instance. There is a kind of spray that is nice for, because in the mucus that is formed in the cystic fibrosis, you have a lot of DNA, because you have a lot of net being released in this disease too, and contributing for the cystic fibrosis. And so they in such a way they help to fluidify to make more fluid the mucus in the cystic fibrosis.
So nice.
Yeah, there are, and there is people also trying to use nets as an adjuvant. How can you imagine to use so many proteins, but you have a lot of toll-like ligands, and you have other
and also NLRP3 activation.
Yeah, sure.
All of this, yes.
They are trying to use this as an adjuvant.
So thanks, Elvira, and thanks, Pamela, to wait for my question before raise your hand.
No problem. I'm trying to figure how can I ask the question. So my question is about the earlier neutrophils. Just mature neutrophils can do NETosis, or the immature neutrophils can do this?
The only thing that I can tell you is that when we isolate neutrophils from bone marrow that you have mature and immature, we can see net release, but anyone could identify if the immature or mature are doing, if one is doing more than the other one. So, and if you look neutrophils from bone marrow, at least from mice, that's easy for you to get, they release nets. But you have a mixture.
Right. And another question is about the agent. If you look the senescences like stage-cell 4, stage-cell 2, you can isolate this population and see that if they release most NETosis than others, or it's not possible. I don't know if flow cytometry or the other methodologies.
As far as I know, anyone was trying to do that. In this meeting that I was in the Paul Cube's talk, he was saying that his lab was engaging in describe neutrophil subpopulation markers. And this was in the meeting that was in Natal. I forgot the year. And till now, no marker is being reported till the moment. So I don't know, but this could be.
Another question is about low density. It's how can I isolate this? It's a little.
It's very easy. It's just to do a fecal gradient, histopath.
Yeah.
And then you have the neutrophils in that the normal ones that will work, and you have much more that are just above the red blood cells layer. And the other one is together with the PBMCs.
Yeah.
The problem is because to have them, you need to get to purify them from the other cells. And in some disease, for instance, in lupus, the people with lupus, they have more low density neutrophils than normal people. So this way is easy to get them. I don't know what is the percentage of the low density in the normal health people. I mean, but
working with rheumatoid arthritis, and I think that could be more low density too.
There is a lot of papers in relating net release with arthritis. Rheumatoid RA.
Perfect.
Yeah, because the nets are being deposited in the joints and doing many stimulations and things there, and exactly why the people with this kind of disease also they release more of these structures. So, but there is a lot of papers about nets and rheumatoid arthritis.
Thank you.
You're welcome. Thank you for the question.
It was an amazing time of discussion. Thanks, Elvira, for being patient and answering all the questions. I think it was really, really great. Thanks for the students as well for participating and engaging the activity. So I think we are done for the day, right, Gustavo?
Yeah.
And tomorrow, what's the schedule?
Yeah, so just a couple of things. First of all, Elvira, if you go to the chat, I added the paper in Cytoplast. And
great.
Try to download. I think we have enough information in the protocol, but if not, then we can talk. And regarding the schedule tomorrow, tomorrow we have one recorded lecture that you can be free to watch whatever time you like it. But then we have the closing lecture at 3, from 3 to 4:30, whatever, from Vishva. So be again 15 minutes, 2:45, everybody. I'm not sure if I added to Moodle or if I sent to you Domagoy's talk. Have I? So you have access.
You sent my mail, and I sent to my students also.
So you have it, so you can you can listen today. You can whatever, okay. So, but the only thing is that you will need to get a summary inside your
Wonderful.
report.
Of course,
Okay.
yes.
So tomorrow we have synchronous, just one activity, right, Gustavo?
Just one
Yes.
activity, quarter to 3 to listen to visual. Okay.
Perfect.
Thank you, Elvira.
Elvira,
Elvira,
it was amazing.
thank you. Congratulations for your work. Fantastic.
Awesome.
Thank you.
All right, guys.
I like very much to work with this glamorous kind of
It is.
Yeah.
Very nice. Nice.
Bye-bye. Thank you.
Great.
Bye-bye.
Bye. Thank you,
Thank you.
everyone.
Bye. Thank you.